All right, let's get our virtual lab coats on because today we are stepping right up to the lab bench. We're going to treat this explainer like a cinematic, hands-on walkthrough of a real laboratory experiment. We've got some fascinating, authentic field notes to guide us, and we're going to perform a classic, absolutely crucial chemical analysis. We're going to determine the acid number of a given oil sample. So roll up those sleeves because we're putting everyday cooking oils to the ultimate chemical test.
You know, it's a question we almost never ask ourselves when we're making dinner. You just reach into the pantry, grab a bottle of oil, and pour it into the pan. But is that oil actually fresh, or is it secretly going rancid? You see, fats and oils break down over time. Today, we're going to look at the exact procedure chemists use to answer that question scientifically. Here's our game plan for today. First, the purpose and principle. Second, our lab setup and materials. Third, the titration procedure itself. Fourth, some crucial lab precautions. And finally, calculating our results. Okay, jumping right into part one, the purpose and principle. The whole aim of our experiment here is to determine something called the acid number. And this brilliantly illustrates the chemistry behind food spoilage. As fats break down, the molecules actually separate, releasing what we call free fatty acids, or FFAs. The principle is pretty simple. We just want to measure exactly how many of these free fatty acids are floating around in our oil. To pull this off, we use a base, specifically potassium hydroxide, or KOH, to neutralize that acid. So the acid number is defined as the exact milligrams of KOH required to neutralize the free fatty acids in just 1 single gram of our fat sample.
So what does this all mean? Well, it's a direct correlation. A high acid number means you've got a poor quality lipid sample on your hands. Think about it. If it takes a massive amount of base to neutralize the oil, there must be a ton of free fatty acid present. And that means the oil has really degraded. It's literally a measurable proxy for freshness.
Moving right along to part two, lab setup and materials.
To do this right, we need to gather a few specific tools for our lab bench. First, our alkali solution, which is our standardized 0.1 normal alcoholic KOH. We also need a specialized solvent mixture to actually dissolve our thick oil sample. Our notes specifically call for a precise 1-to-1 ratio of ethanol to ether, making up a 100 milliliter batch. We'll also need a visual indicator called phenolphthalein, and of course, our glassware, a conical flask to hold the reaction, a burette to dispense our KOH drop by agonizing drop, and a pipettes for precise measurements.
All right, let's get into the action. Part 3, the titration procedure. We have these really cool, authentic, handwritten lab notes guiding us here, showing the exact initial steps. For our sample prep, we start by weighing out roughly 5 grams of our oil and putting it into our conical flask. Then, we add 50 milliliters of that 1-to-1 solvent mixture we just talked about right into the oil. We give it a good swirl until the lipid sample is completely dissolved in the organic solvent.
Next up is the titration itself. We drop in just 2 to 3 drops of our phenolphthalein indicator. Now, right now, the solution is acidic because of all those free fatty acids, so the indicator is totally colorless. We position our flask right under the burette filled with KOH, open the stopcock, and let the KOH fall in drop by drop. The absolute key here, kinetic energy. We have to shake and swirl that flask continuously as every single drop hits the liquid.
As you drip the base in, you're going to see these quick flashes of pink that just vanish as you swirl. But eventually, you hit the climax of the whole experiment, the end point. The lab notes are super explicit here. They say to mix at least 30 seconds to a minute to reach the light pink color. We are hunting for a very faint but permanent light pink tint. The exact moment that pink color stays for a full minute without disappearing, boom. You close the burette, you have perfectly neutralized those free fatty acids. Before we crunch the numbers, let's talk about part 4, crucial lab precautions. The experimenter deliberately dedicated an entire section of their notes just to safety, and for good reason. Potassium hydroxide is a highly reactive caustic substance. This is real chemistry, so it demands our full attention.
Let's break down these non-negotiable safety and precision steps. First, you have to add your indicator just before you start titrating. Second, when you see those pink flashes lingering longer, you must slow your titration down to a crawl. I mean, drop by slow drop. If you overshoot it and the liquid turns a dark magenta, your data is completely ruined. Obviously, avoid any skin or eye contact with the KOH. And here's a wild detail. KOH dilution is exothermic. That literally means it releases heat.
If you're holding the flask, you're going to feel it getting physically warmer in your hand.
Okay, the fun part. Part 5, calculating the results. To turn our pink liquid into actual hard data, we use this specific formula. The acid number equals the volume of KOH we used, multiplied by the normality of our KOH solution, which is just its chemical concentration, multiplied by 56.1. Then you divide that whole thing by the exact weight of the oil sample in grams. Now, you might be wondering, what on earth is 56.1? Well, that's just the molecular weight of potassium hydroxide. This formula perfectly translates our liquid titration volume into that standard acid number metric.
Let's look at the actual math our chemists did. On their calculation page, they didn't just test one oil, they tested 2 totally different samples to compare them: coconut oil and groundnut oil. You can see exactly how they took their burette readings and plugged them right into the formula we just looked at.
And the data paints a really clear picture. The coconut oil sample came out with an acid number of 2.805, but the groundnut oil sample, it needed significantly more base to reach the endpoint, giving it an acid number of 16.83. That is a massive numerical difference. There are clearly way more free fatty acids floating around in that groundnut oil.
But numbers always need context. Are these oils ruined? Let's check our results against typical safe ranges. For coconut oil, the normal range is 0.5 to 8.0, so our 2.805 is looking perfectly fine. For groundnut oil, the typical range is 2.0 to 15.0, so our result of 16.83 is just a tiny hair outside the top end of that average.
But check out the chemist's final verdict. Even though the groundnut oil was running a little high, the conclusion in the notes is crystal clear. Both samples show low to moderate free fatty acid content. The official call is that both are of good quality and completely suitable for consumption. Phew. We can all breathe a sigh of relief. Our hypothetical kitchen is safe, and the oils pass the test.
It is honestly amazing how much hidden chemistry is happening right inside our kitchen pantries. Every single drop of oil carries a specific, measurable signature of its freshness, all dictated by the invisible breakdown of triglycerides into free fatty acids. So the next time you pour oil into a hot frying pan and hear that sizzle, are you going to stop and wonder what its acid number is? Thank you so much for stepping into the lab with me for this explainer. Until next time, keep questioning the everyday science all around you.